Hi, welcome to this video on organisation in plants. Like us, plants are made up of several organ systems comprised of organs made of several tissues, the building blocks of which are cells. Previous videos have looked at the general structure of plant cells as well as some more specialised plant cells. One such organ in a plant is a leaf. The job of a leaf is to catch sunlight to make food in a process called photosynthesis. This organ relies on several different tissues to do this job, which I will go through. Please bear in mind that this picture is a cross-section of a leaf, which would measure no more than maybe a couple of millimetres in width. The upper and lower surfaces of a leaf are covered with epidermal tissue. The upper epidermis secretes a substance that feels waxy and is called a waxy cuticle. This reduces water loss by evaporation from the leaf. But being waterproof, it also redirects rainwater to the soil to be absorbed by the roots. The upper epidermal cells are also transparent. This allows light through to the cells below that, which carry out photosynthesis. The lower epidermis is full of holes called stomata. These let carbon dioxide diffuse into the leaf. Now, carbon dioxide is a reactant required for photosynthesis to occur, so it needs to get in. These stomata are able to open and close in response to water availability. The reason why they can do this is because they are surrounded by two guard cells. The midsection of this leaf is called the mesophyll, which literally does translate as middle leaf. And this is where all the photosynthesis occurs. All cells here contain chloroplasts, which house the green pigment chlorophyll that is able to harness the sun's energy. The upper mesophyll tissue is made up of rectangle-shaped palisade cells. Their brick-like shape means that more of them can pack together just below the transparent epidermal cells. They also contain the highest number of chloroplasts to access and therefore harness as much sunlight as possible. Below this we have irregular-shaped mesophyll cells that are unable to pack as tightly together as the palisade mesophyll, and so they form air pockets like a sponge. This is why they are called the spongy mesophyll tissue layer. These air pockets allow carbon dioxide and water vapour that has entered a leaf to enter as many cells as possible, as there is a bigger surface area where the mesophyll cell membranes meet these two gases. For water to reach the leaf, veins spread throughout the leaf, and these veins are made up of two types of tubes called the xylem and phloem. And it is the xylem tubes or xylem vessels that carry water up from the soil to the leaf, and water diffuses from the xylem by osmosis into these air spaces and then into these cells. The other tube is called the phloem, and this carries the dissolved sugars made by photosynthesis to all other cells in the plant. The following three organs, the roots, the stem and the leaves, together make a transport organ system in a plant, and this system has two jobs. If the phloem is involved, it is involved in translocation, and if the xylem is involved, it is involved in transpiration. Translocation is very simply the movement of dissolved sugars around the plant, and that's all I really need to say on that matter. Transpiration is a much more complex process, and I will cover this in a separate video. So in terms of how you could be assessed on this information in an exam, you could simply be asked to label a diagram that is missing those labels. So my advice to you would be to rewind this video, pause it before I started annotating the diagram of the leaf, and see how many tissue names you can remember. And then to extend yourself, see if you can remember how those different tissue layers are adapted to do their job. Another top tip would be to practice comparing different tissue types. So for example, if you take the upper and the lower epidermal cells, similarly they both lack chloroplasts, they can be transparent and they release this waxy cuticle layer. The difference being is that you will find that there are more stomata and more guard cells in the lower epidermal layer, and the waxy secretions would be less than on the upper layer. Another comparison worth making is between the palisade cells and the spongy cells. They share the fact that they have loads of chloroplasts, but the palisade cells would have more chloroplasts and they would have a more regular shape, allowing them to pack more tightly together than the irregular shaped spongy cells. if you can practice doing those sorts of things, and you'll be effectively revising this subtopic. Good luck and thanks for watching.